This is Fresh Air. I'm Terry Gross. More and more people are looking to genetics to understand where they come from and what their future might be. Genetic sequencing can tell us about our ancestry and warn us about the risks we inherited of certain diseases and conditions. Meanwhile, scientists are exploring ways of altering genes to prevent or cure illnesses. My guest Carl Zimmer has written a new book about state-of-the-art genetic research and the history of genetic research that led us to where we are today. The book is called She Has Her Mother's Laugh: The Powers, Perversions, and Potential of Heredity. Zimmer is a science columnist for the New York Times. He teaches science writing at Yale University. He's written previous books about viruses, parasites, and evolution. Carl Zimmer, welcome back to Fresh Air. How accurate do you think the DNA ancestry traces are now?
I like to think of them as telescopes in the 1700s. You know, they're pretty good. You know, you could look up in the sky with a telescope in the 1700s and you could see things that you couldn't see with your naked eye, but it was pretty blurry, and there might be things you saw that weren't real, and there would be a lot of things you didn't see. So if you go from one testing company to another in terms of ancestry, you're going to get different percentages. People have done this, and that's what they find. The statistics are still very rough. They are going to get better because what's going to happen is more and more DNA is going to go into these databases. And it may even be possible in the future to zero in at least some of your ancestry to, you know, let's say a town or a village in some place where people don't move around a lot. So, you know, you shouldn't look at that as sort of absolute truth. We like to look at our DNA as some sort of divine revelation about who we are. But really what you're getting from these tests is, you know, the best guess that scientists can use with the information that they have.
What did you learn medically from your DNA?
I was working with a genetics counselor when I was getting my genome sequenced. You know, I wanted to know if there was anything I needed to worry about, and I have 2 kids, and I wanted to talk with them about anything that was of serious concern first before even thinking about writing about this. My genetics counselor called me one day and said, "Okay, your genome sequenced, and we've had a chance to look at it." And I said, "Okay." And she said, "We can just have this conversation over the phone," which I thought was weird if she was going to tell me about some terrible, disturbing problem I had that I didn't know about. And she said, "You're fine. I mean, you have a boring genome." And it's funny how I felt somehow a little crestfallen. Like I wanted something exciting and exotic, you know, like I have some obscure, bizarre syndrome that nobody else has, and I'll be cool. But she's pointed out, no, a boring genome is a really good genome. And so all that means is that I don't have any of these mutations that on their own can knock you down with a particular disease. You know, I have, you know, lots of genes with mutations that raise my risk a little bit for this disease or that, but we all do. What was actually really interesting to me is that when I showed my genome to some scientists, they said, you know, you actually have an interesting mutation here in this one gene. This isn't a gene that raises your risk of disease. It is actually extremely protective. So I have a mutation that protects me from certain autoimmune diseases. And when I started to learn about what this mutation does and how it sort of tamps down my immune system so it doesn't go out of control, I discovered that scientists have been studying this mutation in other people and have developed a drug based on the biology, which has just gone on the market recently and is used for autoimmune diseases. So there's no end of these sorts of things to discover. You know, I can look to my parents and say, you know, there's a mutation I inherited from each of you on a gene called FTO, and you know that makes people on average a few pounds heavier. So you know, I wish you had given me the other one.
No, but they gave you the great one, the one that protects against autoimmune disease.
That's true. Well, one of them did. I have one copy.
But that's good enough, isn't it? No?
Yeah, it is. I don't know which one personally to thank, though.
A question in terms of heredity has always been like nature versus nurture. Like what potentials and problems are you born with because they're inherited through your genes, and what's going to shape you from the world around you, the experiences you've had, the environment you're brought up in, the way your parents raise you. And you say that line is growing increasingly more blurry, and that's in part because of our knowledge of epigenetics, which is a whole new field about how parts of your genetics can actually be changed by the world around you. Would you explain the basic premise of epigenetics?
Sure. So we have genes, and our cells use genes to make proteins and other molecules. And those genes can be switched on or they can be switched off. And the way that happens is that our cells have molecules that are hovering around our DNA, and they clamp onto certain genes and silence them. We can actually coil up our DNA to hide genes, and that shuts them down as well. And these are long-term changes, so that when an embryo is developing and starting to develop muscle and brain tissue and different organs, the cells in each of those tissues are permanently turning off certain genes and turning on others. And that whole area of turning genes on and off is sometimes called epigenetics. And what's fascinating is that actually it looks as if some kinds of experiences that we have in the environment around us may alter that epigenetic pattern in our cells. So it's conceivable that in your own life, your experiences can change how your genes work. This is a very controversial area for a lot of reasons. One is that it's actually surprisingly hard to really pin down what those changes are and what's causing them. But that hasn't stopped epigenetics from becoming incredibly trendy. And where this gets even more exciting but also controversial is the idea that maybe if your experience changes that epigenetic signature, maybe you could pass it down to children, and maybe they could pass it down to their children. So perhaps some people argue epigenetics could be a completely separate channel of heredity.
So there was a study related to epigenetics that was done with mice. And what they did was they totally stressed out mice to see if that stress was inherited by the mice offspring. Would you describe that study and tell us how definitive it is? Like what did we really learn from it?
These studies are really fascinating because what they're doing is they're putting mice under stress, male mice in particular, and then these mice are mating, and then researchers are looking at their offspring and later descendants. And you have to think about this, that all the male is contributing to its offspring is just sperm. It's not as if females are getting stressed and that affects the environment in the uterus where an animal is developing. So
the stressed out mice were just male mice.
That's right. So it's just male mice, and all they are contributing is what's in that sperm cell. And some of these experiments suggested that their descendants seem to be altered through the experiences of their father or grandfather. And then scientists have actually gone into these individual sperm cells, and they have pulled out some of the molecules that control genes. And they've actually then just taken those molecules and then gone over to some other mouse sperm and inject them into that, and then use that to fertilize mice. And just transferring those molecules into other mice seems to then produce these offspring that also have this kind of altered personality. This is incredibly tantalizing research. It makes you think about how the experiences of our ancestors might alter us. But a lot of scientists are really strongly skeptical about it. They are arguing that these are very small studies, that they're just very random effects that are being picked up, and people are claiming that they're real. So, the jury is definitely still out on mice, let alone people. But, that doesn't mean that epigenetics isn't real.
Let me reintroduce you. If you're just joining us, my guest is New York Times science columnist Carl Zimmer, who's the author of a new book about the history of genetic research and current state-of-the-art research. The book is called She Has Her Mother's Laugh: The Powers, Perversions, and Potential of Heredity. We'll be right back after a break. This is Fresh Air. This is Fresh Air, and if you're just joining us, my guest is Carl Zimmer. He's the author of a new book about the history of genetic research and current state-of-the-art research. It's called She Has Her Mother's Laugh: The Powers, Perversions, and Potential of Heredity. And he's a science columnist for the New York Times. Let's talk about CRISPR, which is the word that's used for the process of what's called genetic splicing, like genetic editing, basically changing a gene. And, when you hear genetic splicing, at least for me, I work in radio, I picture like audio editing, audio splicing, where in the old days we would remove a piece of tape and splice the remaining pieces of tape together, or now digitally edit out a few words or a few sounds. But CRISPR, from what I understand, isn't anything like that.
Actually, I think that's not a bad metaphor.
Really?
Yeah, because what happens with CRISPR is that scientists will design a molecule, think of it as a probe, and it will search around in the DNA in a cell until it finds a very specific short sequence, and it'll grab onto it, and it brings along with it basically molecular scissors, which will then cut the DNA at that spot, kind of like cutting tape. And you can cut out a segment of DNA, and if you just do that, then the DNA will heal itself. Basically, the two loose ends will stitch themselves back together, and now that piece is just missing. Or you can add in a little piece of different DNA, and you can actually get the cell to put in that new piece of DNA where you just cut out the old one.
Right. So that's pretty remarkable. But it's not like scientists are like taking razor blades or anything in the lab and changing DNA. You're basically programming other molecules to go in and do the job for you?
Yeah, you're creating molecules that are going to be able to attach to just one particular place in all of your DNA. And so they are zeroing in with greater and greater precision to just particular spots in the DNA. Wherever you want to go, you just craft a molecule that will recognize that place and lock onto it. And then it brings with it these molecular scissors that will make the cuts.
I'm confident that if I asked you to explain it in more scientific detail, that I would not understand it. So instead of doing that, I'm going to ask you for an example of how that's being used now, how this kind of gene editing is being used.
So here's an example which I think really drives home the potential for CRISPR. So scientists can create lines of mice that have some of the diseases that we have. So, for example, there is a kind of mouse that gets a muscle disease called muscular dystrophy. And this is caused by a particular mutation on the X chromosome. And it's a really devastating disease in humans, and these mice will develop it too. So you have these mice that, unless otherwise treated, are going to basically waste away. Their muscles are essentially going to turn into like a jelly-like substance, and then they're not going to be able to breathe anymore, and they're going to die. Now, what scientists can do is they can inject these CRISPR molecules into the mice. And these CRISPR molecules make their way into muscle cells, and they then cut out that mutation and repair the DNA. So the mutation that caused the disease is no longer there in those cells. And these mice then become stronger, and they live longer. They are being treated, if not cured, of this disease. That's the kind of thing that CRISPR can do.
That's pretty remarkable. Is it being used with human beings yet?
We're just on the verge of human trials. They will be starting hopefully very soon for diseases like sickle cell anemia. There's actually a lot of research on muscular dystrophy as well. There are a few key diseases where scientists think these would be the best places to start, to basically inject CRISPR molecules into people's bodies. These CRISPR molecules will then go to certain kinds of cells and repair one particular spot in their DNA, and that treats the disease.
So this could be the future of genetic diseases, genetically inherited diseases.
It very well could be, yeah. I mean, we shouldn't look at this as a panacea because it may turn out that these CRISPR molecules, maybe they don't do a very good job of reaching their targets in the human body. Or maybe there are going to be side effects like, for example, maybe they get distracted by another piece of DNA and they accidentally cut that as well. I mean, there are a lot of reasons to sort of just kind of hold on because it's so exciting that you want it to work. But we've been here before. I mean, there have been earlier kinds of treatments known as gene therapy where you would basically try to add an extra gene into someone's cells. And that seems like it was just a slam dunk, but then it turned out to not work very well for years and years, and that's only starting to recover now after like 20 years of research. So CRISPR could be even more exciting and truly revolutionary. We just have to wait and see what these first generation of human clinical trials show us.
Is CRISPR being used for genetically modified foods, or is that a completely different process?
They are being used for genetically modified foods. They're just beginning to. And the basic logic is, again, the same in the sense that, plants have DNA and CRISPR works on DNA. So what scientists are doing in that case is they're not just trying to, say, cure a disease in a plant. They're actually trying to produce new breeds of plants. And so, if you know that there is a gene that controls how a plant deals with high temperature, and you're saying, well, we need to prepare new kinds of plants for climate change. You can test out new kinds of genetics by using CRISPR to rewrite that gene a little bit and see how well it does at those higher temperatures.
There are some people who will not eat genetically modified food if they can avoid it. And I'm wondering, after having done all this research for your new book, what your thoughts are about the pros and cons of genetically modified food.
Well, the fact is that, when plant breeders were producing new kinds of plant varieties, like in the 1900s, they would use methods like basically pointing X-ray machines at seeds and just blasting them, and then just seeing if any interesting mutations came out of that, and then breeding up some crops from that. That's like a very random, almost blind process, and so there's no reason to think that actually going in strategically and tinkering with one particular gene that you already have a lot of evidence is very important for the trait that you want to change could be any worse. In fact, you might have good reason to think that it would actually be safer. There's no evidence that genetically modified foods are harmful to our health. And so there's even less reason to think that this new generation of CRISPR foods will necessarily be more dangerous to us. We may object to genetically modified foods for other reasons, for social and economic reasons. And I find those arguments to be totally valid in the sense that, do we want to leave the control of agriculture in the hands of small-scale farmers, or does everybody have to go to, one big chemical company and buy their seeds each year from these people and be not even allowed to plant those seeds the next year? That's a different argument, though. That's a social argument, and it doesn't have to do with safety.
There's also a fear that the genetically modified plants will spread their DNA to places where that genetic modification is not wanted, and that they'll just kind of like take over?
There has been concern about that, but there's no evidence of that actually happening in any sort of significant way. So genetically modified crops have been around now for a number of years, and in the United States, a lot of the the soybeans and cotton and so on these plants are genetically modified, and you don't see this sort of runaway process happening.
My guest is Carl Zimmer. His new book is called She Has Her Mother's Laugh: The Powers, Perversions, and Potential of Heredity. This is Fresh Air. I'm Terry Gross, back with New York Times science columnist Carl Zimmer. His new book, She Has Her Mother's Laugh: The Powers, Perversions, and Potential of Heredity, is about state-of-the-art genetic research and early genetic research that led us to where we are today. When we left off, we were talking about research into how genes can be modified to prevent diseases. So another area that applies to modifying genes for genetic reasons to prevent disease has to do with genetically modifying insects that carry disease, such as trying to genetically modify mosquitoes so that those mosquitoes can no longer carry malaria. What's being done in this research?
So for the research for my book, I went to California and went to the lab of a scientist named Anthony James, and he runs what he calls an Insectarium, which is basically a giant room full of mosquitoes. He raises thousands and thousands of mosquitoes, and he's genetically engineered them so that they are resistant to malaria. Malaria is spread by a parasite, and so these mosquitoes basically kill off the parasite if it tries to get in their bodies. And the idea behind these mosquitoes is that maybe in the future we would release them into the wild. And these mosquitoes have been genetically engineered in another way so that they will basically push this resistance gene into all of their offspring. And those offspring will push it into all of their offspring as well. So they basically override the normal rules of heredity. And the theory is that if you were just to release a few hundred of these mosquitoes into a region where you have a lot of malaria, within a few years, the malaria might be gone. This could be a way of eradicating malaria, period. And so now there's a huge amount of interest in this from a lot of foundations and governments around the world focusing on using this new way of tinkering with heredity to maybe attacking malaria.
Is there a possible downside of doing that?
The downside is that once you set these kinds of organisms loose out in the wild and you kind of change the rules of heredity, it might be hard to undo what you've done. We've sort of seen an analogy of this with invasive species, that people brought cane toads from South America to Australia because they thought they would be great at eating the insects that were destroying sugarcane plantations. And it turned out the cane toads like to eat lots of native species, and they've exploded over the continent, and there's just no way of taking them back. So maybe these CRISPR-altered mosquitoes might do something that we can't predict, and they're just going to keep reproducing and just be part of the environment. And we really don't know much about how to take them back. So there is a lot of research going on into how to make this gene drive technology super safe and being able to undo any damage that you might do to wild species.
So the Insectarium that you went to with all the thousands and thousands of mosquitoes, would you describe it to us?
So yeah, it's kind of amazing. I mean, first of all, you have to gown up before you go in there.
That sounds like a good thing.
Yeah, absolutely. But it does sort of remind you you're doing something serious here. And then you go through an airlock, and then you're in this room where there are the mosquitoes living in all their different life cycles. So there is a dark room where the female mosquitoes are laying their eggs because they like to do it in the dark. And then the scientists pull the eggs out from these rooms and they inject DNA into them. And then they put them in water because that's where mosquito larvae like to develop. And so you go into this other room where there are these tubs of water and these like snake-like things are slithering around in there. And then they develop into adults. And then, the females need to drink blood. And so they have these, they found that the containers for movie popcorn work really well. What they do is they basically clamp a warm container of calf's blood on top of them, and then the mosquitoes are underneath on the underside of the plastic lid, basically poking through and drinking the blood and fattening themselves up, and then the cycle repeats itself. It's kind of spooky, and when you leave, you go back into the airlock, and then you just look at this white door, making sure that no mosquitoes came out with you, because for now, those mosquitoes need to stay in there. We do not want them getting anywhere. It's a very intense experience.
So when you were in the room all gowned up, were the mosquitoes wild, or are they contained like in an aquarium where everything's behind glass?
The mosquitoes were always in containers, so they were, they were in jars, or they were in these little plastic tubs if they were still in their larval stage. So no, you were not getting bitten by mosquitoes. They were not swarms of mosquitoes in your face. They were all very much where they needed to be in different rooms for different parts of their life cycle. But it's always possible that one mosquito might get out, and they have all sorts of things in place to basically kill any mosquitoes that might escape. These are actually mosquitoes that are native to India, and this is Irvine, California. So even if one were to get out, it would probably just die right away because it's so dry there. It's so unlike their native habitat. So all sorts of things have been put in place to minimize the chance of anything going wrong.
And you didn't have any bites when you left?
Nope, I did not. But the flip side is, there is certainly it's a little spooky to be looking at, movie popcorn containers full of blood engorged mosquitoes. But on the other hand, you look at them and actually you can tell that they've been genetically altered because they have red eyes, which is kind of spooky. But, you look at that and you say, well, that means that these could be the cure for malaria. They really could happen. And hundreds of thousands of people die every year of malaria. We've thrown everything we can at it, and this parasite is still knocking us down worldwide. So maybe this could be it. And so that's actually quite exciting.
Let me reintroduce you here. If you're just joining us, my guest is science journalist Carl Zimmer. His new book is called She Has Her Mother's Laugh, The Powers, Perversions, and Potential of Heredity. And it's about the history of genetic research and the state-of-the-art current genetic research. We'll take a short break and then be right back. This is Fresh Air. This is Fresh Air, and if you're just joining us, my guest is Carl Zimmer. He's a science columnist for the New York Times and author of a new book about genetics, the history of genetic research and current state-of-the-art genetic research. It's called She Has Her Mother's Laugh, The Powers, Perversions, and Potential of Heredity. So one of the big stories lately involving genetics was how genetic research, how DNA was used to track down the Golden State Killer in California. And this had been a cold case for years. So how is DNA used to solve that case?
They were taking advantage of a pattern in heredity, which is that, as parents pass down DNA to their kids, the DNA on the chromosomes gets shuffled a little bit. And so what that means is that, you and a sibling will share a lot of very long identical stretches of DNA, but you and your cousin will share fewer of these sequences, and they'll be shorter. That's because there's been more shuffling happening over 2 generations instead of 1. and the same is true for 3 generations back, and so on. So geneticists have actually been able to come up with a method for comparing DNA, identifying these long stretches and saying, these two people must be related. So this method of identifying relatives has become incredibly popular thanks to the rise of websites like Ancestry.com, 23andMe, and they'll offer you this relative finder service. And basically all they're doing is looking for people in their database with very long stretches of DNA that match yours. And so for the Golden State Killer case, what somebody decided to do was to take the DNA that they had from these crime scenes and upload it to one of these open access sites, not a commercial site, and then just see if they could find any close matches. And they found that there were some people who looked like they were distant cousins of this person. And then they went and did the genealogical research to figure out, well, how would they be related? And then said, okay, who are the possible relatives that this person could be and where do they live? And that actually helped narrow down their search until they made an arrest.
Now, if you just send your DNA to get like an ancestry search through one of the companies like 23andMe, would that mean that your DNA was available for a crime search like that?
So the commercial companies said when they were asked about this during the Golden State Killer news that they won't do this. I mean, that they will not invade people's privacy in this way. Now, what the police did in this case is that they went to an open access sort of crowdsourced site called GD Match, and there basically, it's sort of a group effort, and everybody uploads their DNA and works on the software together, and it's sort of a joint effort. And so, everybody is free to just try to match up their own DNA to anybody in the database. I don't know how many people thought about the police coming in and secretly comparing their own DNA to a possible suspect's. This is a broad civil liberties issue. There has been some long-running concern about police trying to find evidence of crimes by looking for the relatives of criminals with DNA because, things get less and less precise. And there have actually been people who have been arrested because it looked as if, their DNA matched that of somebody in one of these genealogical databases. Like they looked like they were related, and they said, "Oh, that's good enough for us," and they would make an arrest. And then it turned out this person was innocent. So this is not the last time we're going to see this coming up in news about crime.
Well, getting back to the commercial companies like 23andMe and Ancestry, what kind of privacy agreements do you sign with them?
So you can choose sort of different levels of privacy with a lot of these services. And so, for example, some people will say, I want you to look at my DNA. I want you to tell me about my ancestry. I want you to tell me about, for 23andMe, they'll give you a few bits of information about your medical conditions, and that's it. But they will try to get you to opt in to sharing your data for their own basic research. So at 23andMe, for example, there's a whole team of researchers who are studying all sorts of things, all sorts of diseases, sleep patterns, and so on. And then they will also go into partnerships with drug development companies who will take their data, looking at, say, 50,000 people with lupus and 50,000 people who don't have lupus, and try to look for the genetic differences. Those could point the way towards possible drugs.
So to sum up, there's some amazing breakthroughs being made in genetic research, but there's so many questions that remain. I know I'm stating the obvious here, but it sounds like there's a lot that's still really inconclusive.
Yes, if you just look at the genome and look at all the things that we inherit genetically from our ancestors, there's just a lot of it that scientists really can't tell you much about at all. It's still a pretty poorly explored frontier, and heredity is so important to us. It really is how we explain who we are and how we got that way by looking to our ancestors and saying, what did we inherit from them? And so we really want it to tell us all sorts of things. And now that we can start to look at our own DNA, we want those answers right now. And the simple fact is that a lot of those answers either aren't there yet or we'll never find them in our DNA.
So when you decided to have your genome sequenced, and that was really part of the research for this book, because you knew you'd be writing this book, had you braced yourself for the possibility of bad news, that you would find out that you had a gene that showed you would be more inclined to have, say, early onset Alzheimer's or an inherited illness?
I was definitely concerned. My father's parents both died pretty young, and in both those cases, they were diseases, heart disease in one case and cancer in another. And so I thought, oh boy, like I could very well have inherited whatever genes that put them at risk. And my father is Ashkenazi Jewish background, and so I immediately started thinking about this gene that people may have heard of called BRCA1, which is a gene that, if it's mutated, can really raise your risk of breast cancer. I have 2 daughters, and so that got me very anxious as well. So yeah, I would say that it was a very nerve-wracking experience going into it. I think we all think back to our relatives who got sick and then wonder, well, is that in me? I mean, I have someone else in my family who had an intellectual disability, and I thought, well, is that inherited? I mean, you just keep thinking about these issues, and I wasn't actually going to write about my genome if something turned up, and I was going to personally have to deal with a serious medical condition, or have to talk with my daughters about it. I mean, I wouldn't want to sort of, kind of air their genetic laundry, as it were. But I have lots of genes that slightly increase my risk of some things and decrease my risks of other things, just, nudging the needle a little bit. And that's pretty typical. It's only a pretty small fraction of people who will get their genome sequenced and discover there's something really serious that they have to attend to. And that could be a lot of people. I think there could be a lot of value to people having their genome sequenced in general. But I think any one person, if they really have their fingers crossed that they're going to find some weird exotic mutation, may be disappointed. And that's a good thing.
Carl Zimmer, a pleasure to talk with you again. Thank you so much.
Thank you. It's been great.
Carl Zimmer is the author of the new book, She Has Her Mother's Laugh: The Powers, Perversions, and Potential of Heredity. Fresh Air's executive producer is Danny Miller. Our technical director and engineer is Audrey Bentham. Our associate producer for digital media is Molly Seavey-Nesper. Roberta Shurock directs the show. I'm Terry Gross.